Hello, everyone. Welcome back to Science in the Age of COVID-19 seminar series. As always, we are extremely happy to have you all with us here today. Well, vaccines. Isn't it a buzzword we all want to hear at this point? Well, thanks to the heroic efforts of our fellow scientists, we are making some progress here. We just heard back from 2 clinical trials that appear to look promising. But we still have questions. What does the efficacy look like? What does the safety profile look like? If everything looks good, how do these vaccines compare with each other? When will the vaccine be available? In other words, how close are we to getting ourselves vaccinated and hopefully going back to our pre-COVID lives again? What are the roadblocks along the way? So clearly we have a lot of questions. And when you have questions, you ask an expert. So we are extremely happy to welcome back Dr. Florian Krammer to our seminar series today. Now, I say welcome back because Florian was here with us, I want to say roughly a month or so ago, where he delivered a very insightful talk. So if you missed that talk, please go back, check our recordings, because that is a must-watch talk. For those of you who missed him, I'm going to reintroduce him real quick. Florian is a professor of vaccinology in the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. Many feathers to his cap, but probably the most significant one at this time is that his lab was among the first few labs to develop, optimize, and distribute a robust serology assay to detect an immune response against SARS-CoV-2. And today we are extremely thankful to Florian to take time off of his busy schedule to come back and talk to us. Now here's what we do today, slightly different from the regular Science in the Age of COVID-19 talks. For the first 30 minutes or so, Florian would talk and provide us a big picture overview of vaccines, compare the 2 clinical trials that we heard about, clarify some concepts, and provide us insight. And then for the next 30 minutes or so, we would open up the floor for questions from the audience. We do this all the time, but this time it's going to be a longer and a more interactive one. So I suggest as Florian delivers his talk, please go ahead, start typing your questions in the chat box so that we can get to them right after the talk. This should be fun. So before I start talking and hand over the mic to Florian, I want to take a quick minute to give a big shout out to our AV crew, Matt, John, and Alex, who's a part of the team but is not here with us today. These are the guys who make it happen week after week, every week. Thank you, guys. So with that, Florian, welcome back, and you're all set.
Thank you very much for this nice introduction, Sarada, and thanks for having me back. So what I'm going to try to do for the next 30 minutes or so is to give you an overview of where we stand with vaccine development, and then I hope there's enough time so that people can ask questions that they might have about where we are with vaccines and when they will become available. So I just wanted to show you first a very depressing slide, which is basically a schematic of regular vaccine development. So what we usually do when we develop vaccines. And this usually happens at a very glacial pace. So you design your antigens initially, you do exploratory preclinical experiments. This is often done in an academic lab, and this can take years. And then you go usually with a commercial partner. You think about how you actually can produce the vaccine in the right way. You do more formal preclinical experiments and toxicology studies, and we have just been through that with the flu vaccine. It takes about 2 to 4 years. Once you have GMP material, so good manufacturing procedure material, which is needed to go in a clinical trial, you can submit an IND, so investigational new drug application to the FDA. And then typically you go into a phase 1 where you first look at safety in usually less than 100 individuals. You might get some immunogenicity data. If that looks good, and if your commercial partner is interested and wants to take the risk, you go a little bit further into phase 2, where you have typically a few hundred people that you test your vaccine in. You look at different vaccination regimens. you look again at safety and a lot of immunogenicity this time. It takes about 2 years. If the results look good, then the commercial entity developing that vaccine needs to decide if they take the big risk of starting a phase 3 study, which is very, very expensive, where you actually look at efficacy and safety again of your vaccine in a population of a few thousand individuals. And once that data is in and looks good, you put a dossier together and you submit a biologics license application to the FDA or the EMA if you're in Europe or other regulatory agencies. And then they review that, and that process can take quite a while as well. And then once you're done with that, and once your vaccine is licensed after about 15 years or longer, you actually go and produce it large scale. You wouldn't do that beforehand because it's a big risk to do that. And so we don't have this time right now. And so what emerged was a very modified schedule. So the good thing here is that we know from experiments with SARS coronavirus 1 and MERS coronavirus that we have to focus on the spike protein. We know how we can design vaccines that induce, at least in animal models, very good immune responses against the spike protein. And then partially these constructs already in January and February were moved into process development, and there were already pre-existing processes in some cases. For example, if you look at Moderna, they already had a nice process to make their messenger RNA. They didn't have to change that. And so that sped things up as well. And then basically what happened was that people started with phase 1 trials, but they did adaptive phase 1/2 trials. And for example, you would have to wait, or you would usually wait until the trial is over for safety reasons and so on and so forth, for getting all the data in before you start a phase 3. But basically, these are starting now in parallel. And what the really huge advantage is, or huge time sparing step here, is that the vaccine producers are starting to produce vaccines at risk. That's something you would never do if you do regular vaccine development. And so this makes it possible to actually submit a BLA within months instead of within years. And then there might be some ways of speeding up regulatory approval as well. They could review on a rolling basis. And there's also the question of an emergency use authorization that's floating around. This has been done for a vaccine before, but the situation was very different. However, there are some, although we are moving very fast, there are a lot of issues to keep in mind. The first one is that large-scale production is difficult, and most of the front runners, or 2 of the front runners now in this race, have never produced a vaccine for the market, and that includes Moderna and Novavax. They have no vaccines that are licensed, so they have never produced a vaccine at scale. And a lot of the other technologies that are used are new technologies that have never resulted in a licensed vaccine either. Even, for example, Pfizer, which has licensed vaccines, has never brought an RNA vaccine to the market. So there could be a lot of complications there. Distribution is an issue, specifically because we're talking about large numbers of doses. This takes time. And again, we are talking about millions and millions of doses in the US and billions of doses globally. Administration has some question marks. First of all, we don't know who will get the vaccine first. This needs to be figured out. And then how can you vaccinate so many people in a short amount of time? So the question is, are we going to have mass vaccination campaigns, or is this going to be handled differently? And then in the end, you also have to take into account that you don't get instantaneous protection. You might have to take at least 2 shots. So it might actually take a few weeks to almost months until you mount protective immunity, if we get to protective immunity, which we don't know yet. And so just to, again, highlight the scale and the scale of the problem here, CEPI just put a tweet out saying that they think they have secured production capacity for about 2 billion doses by the end of 2021, Now, globally, we need 16 billion doses. There's 8 billion people, and it's unlikely that one-shot vaccines work. So we need double that. And that will take a long time to get there. And in the US alone, if you think about double doses, actually that number is wrong, we need 660 million doses. So a lot of doses are needed. It's almost unimaginable how much vaccine we'll need. And so I just wanted to walk you through the platforms initially that are used now for making these vaccines. Again, many of those are new that have not been used for vaccines on the market or only in certain instances. Let's start with the RNA vaccines, which are developed by, for example, Moderna and Pfizer. Those vaccines target the spike protein. They have a lot of advantages because you don't need infectious virus in the process of making them. But in the past, there have been safety issues in terms of reactogenicity. DNA vaccines have the same advantage. You don't have to handle infectious virus. The scale-up is easy and you can relatively cheaply make them, but they only work well if you deliver them with specific delivery devices like electroporation devices. And so that makes it a little bit more challenging. Then we have recombinant protein vaccines, which have been used for influenza, HPV, also hepatitis B virus. And so they have the same advantage. There's no infectious virus in the production process, but there is a limited global production capacity for those. And this always depends on the yield of the protein. And we know that the spike protein doesn't express very well, the RBD does. So there's many different variations there. Then we have vectored vaccines, which could be viral, live or replication active vectored vaccines or non-replicating vectored vaccines. And so there's only one of those on the market, and that's the Ebola vaccine, which is called Ervebo, and that's based on vesicular stomatitis virus. So the complications there are that specifically if you have a replication active vector, it could be problematic in people who don't have good innate immunity, who are immunocompromised. And there is also issues with vector immunity in some instances. For example, if you use a common human pathogen or common human virus as virus vector. Then we have live attenuated vaccines, which have been developed for SARS-CoV-1. The process to make them, to design them is not so straightforward, but they are relatively easy to make. And we know how to make live attenuated or produce live attenuated vaccines. But again, the question is, and we have seen that now, it's not so easy to make these live attenuated vaccines. What I mean is designing them and rescuing these viruses. And then there's also safety questions here, of course, with live attenuated virus. And then we have inactivated vaccines, which are very simple to make. We know how to make them. There's a lot of inactivated vaccines that have been licensed and that work very well. There's a lot of production capacity globally for those. The problem here is that this is a biosafety level 3 virus, and there's very few facilities that can actually grow this virus. And there's additional potential issues with antigen or epitope integrity after inactivation. And there's also some concerns about antibody-dependent enhancement with inactivated vaccines, since this has been observed for SARS-CoV-1 in animal models. So those are the basic designs that are in the pipeline right now. So if you look at preclinical candidates, it's amazing how many candidates there are. So this is basically captured by the WHO. We have 140 candidates in preclinical development. The vast majority, 50, are protein-based vaccines, and I lump together here RBT-based vaccines and spike-based vaccines. We have non-replicating vectors. Most of them are adenovectors, but there's also others in there. We have replicating vectors, also replication active adenovectors, VSV, for example, but also measles. There's a number of RNA vaccines, virus-like particle vaccines, DNA vaccines, and what's very interesting, there's actually in the preclinical pipeline very few inactivated vaccines compared to the other categories, and very, very few live attenuated vaccines. And I think, again, this is one of the issues there, is that they're really hard to design and to rescue. I can't go through all the 140 candidates here, but I wanted to go through 4 that I think are worth mentioning. One is a live attenuated approach that's developed by Codagenics together with Serum Institute of India. And this is going to be attenuated by codon de-optimization, which is a very good and safe approach. I wanted to highlight because I think we need a live attenuated candidate, because those can really induce mucosal immunity. I also wanted to highlight it because Serum Institute of India is on board. And in the Western world, we might not hear much about Indian vaccine producers, but Serum Institute is one of the vaccine producers with huge capacity to make vaccines. Then what I also wanted to mention is a spike protein vaccine that's made by Sanofi Pasteur. And I wanted to mention that because that's basically made in the process that is used for making Flublok, which is a recombinant HA vaccine. And they have made a SARS-CoV-1 vaccine that went into preclinical studies many years ago, and that was pretty successful. There's a measles vector vaccine that is produced by Themis, and Themis recently got acquired by Merck. And that's based on a vector that was licensed from Institut Pasteur. And the reason why I'm mentioning this here is because they have the same vector, but with chikungunya in phase 3 studies. And so they could be very quick, specifically with the backing of Merck, to move that ahead. And the chikungunya data really looked very well. And then I also wanted to mention a Newcastle disease virus vaccine that expresses the spike protein that we are developing here, which can actually be produced in the same production process that's used to make flu vaccines, which also is an advantage because there's a lot of existing infrastructure. If you look at candidates in the clinic, there's already 22 candidates in clinical trials. 4 of them are non-replicating vectors. 5 of them are inactivated vaccines. We have 3 RNA vaccines, 4 DNA vaccines. 2 are based on the RBD, which is the receptor binding domain of the spike, and then 4 are based on spike protein. And of those 22, we already have 6 that are have been advanced into phase 3 or are advancing into phase 3 right now. 2 of them are in phase 2. 10 of them are in phase 1-2 studies. So these studies where you actually do a phase 1 and then right away start a phase 2. And then we have 4 that are in phase 1 studies. So this is an astonishing number of vaccines in clinical development, not even a year after the virus was discovered. So this it's just amazing to me. And if you look at which countries these vaccines are coming from, 7 are developed in China, 5 in the US, 3 in Europe, 2 each in India and Australia, and then we have 1 candidate in Russia, Korea, and Japan. And before I now go into the vaccine candidates that I wanted to discuss, I wanted to mention correlates of protection. So for many of the vaccines that we actually have developed, we have correlates of protection, and very often that's a certain type of antibody that you measure. And then you basically determine through studies a titer that is protective. If you're above that titer, you're protected. If you're below that titer, you're not protected. We have that for Hep A vaccines, for Hep B vaccines, for flu, for measles, and so on and so forth. That's very helpful to monitor how well a vaccine works. Now, of course, we don't have that for SARS-CoV-2 yet. My lab is working on developing something like that. Many other labs are working on that. But what we have is, and that explains why we are very often looking at antibody responses in these vaccine trials, we have some idea about what protects non-human primates. And what I'm showing you here are 2 very elegant studies from Dan Baruch's lab in NHPs. On the left side is a study where he infected initially animals in a primary challenge, and then he re-challenged, and he showed that it's very hard to reinfect these animals. And on the right side, he did a similar study where he actually vaccinated animals with a homemade DNA vaccine that wasn't that great, but it induced some neutralizing activity. There were different constructs, so you got a heterogeneity of neutralizing antibody titers. And so he did an analysis of all kinds of immune responses, CD4, CD8, binding titer, neutralizing titer. And very clearly in these non-human primates, neutralizing antibody titers emerged as correlate of protection. So while other measurements did not. So we have already, at least in the non-human primates, a pretty good idea of what we need to look for. And that's why the neutralizing antibody is also going to be the focus of many of the human vaccine trials. Now, another question, and that's very, very important, is what type of immunity do we want to achieve? Do we want protection from infection, which is really hard to do? Do we want to have protection from disease, or at the lower bar, protection from severe disease? And that also has to influence how we design vaccines. The upper respiratory tract is mostly protected by secretory IgA1. And if you want to get protection from infection, you probably have to induce a lot of those antibodies. But that's not going to happen with an intramuscular injected vaccine. What you usually get when you inject the vaccine is a lot of IgG, and IgG protects the lower respiratory tract. That doesn't mean there's no IgG in the upper respiratory tract, but it's going to be really hard, and it's going to require very high titers to protect from infection with an injectable vaccine. And I'm just putting that out there because there's a lot of different expectations, and a lot of people want to see protection from infection, but may not realize that that's very hard with the vaccines that are in the clinic right now. And so what I did is, for the ones that are in the clinic, I just summed up here the data in non-human primates. And then I did this because that gives you an idea about protection from infection versus protection from disease. And so we have our vaccines here on the left, the type of vaccine, the dose that was used, and then neutralizing titers after the prime, the boost, or a second boost, which was actually done by Sinovac with an inactivated vaccine. Then if a T-cell response was found, the challenge dose, which is not the same for all studies. You see 10 to the 6th for the Sinovac study, given intratracheally, for example. Novavax, for example, used 10 to the 4th intranasally and intratracheally, and the outcomes are also a little bit different. So if you walk through them, Sinovac gave this inactivated vaccine with alum as adjuvant 3 times. They had different doses, and they got into, I would say, medium to low range neutralizing antibodies. They challenged through the intratracheally, and so they got lower respiratory tract protection, so no virus replication in the lung, but they didn't test what happens in the upper respiratory tract. AstraZeneca, which is the Oxford vaccine, basically it's an adenovirus that expresses S, was tested as prime or prime boost, and they got titers initially at in the 1 to 10 range, and then it went up to the 1 to 100 range after the boost. When they challenged, they got partial or no protection in the upper respiratory tract, but full protection in the lower respiratory tract. Again, infection, but no disease. Janssen tested an Ad26 vector that also expresses the spike. They got into the 1 to 100 neutralizing titer range after 1 shot. And again, when they challenged, they got very nice protection of the lower respiratory tract, but they had some signal in the upper respiratory tract. Only in 1 animal, I believe, but there was some replication. Moderna just published their study. They gave 2 times their RNA vaccine, which is formulated in LNPs. And they achieved relatively high neutralizing titers, 1 to 100 to 1 to 1,800, depending on the dose. And again, when they challenged complete protection of the lower respiratory tract, in the low doses, they didn't see good protection of the upper respiratory tract, but they saw very little replication at the high dose. So that looks pretty good as well. And then Novavax just came out with a press release recently describing their spike protein-based vaccine with a saponin adjuvant, matrix M, and they found a really, really high neutralizing activity after the boost. And what they reported after 10 to the 4th DCID50 challenge, so much lower than all the other ones, they reported complete protection in the lower respiratory tract and protection from infection. So that looks like sterilizing immunity, but keep in mind the challenge dose was lower. If we go into the clinical trials, we have seen data from CanSino. So CanSino is a Chinese company that worked together with the Chinese military. They developed an adenovirus 5 vector that expresses spike. The data that they published in the Lancet is based on their phase 2 trial. They're currently in phase 3. They gave 2 doses, 5 times 10 to the 10th and 10 to the 11th virus particle in a single shot intramuscularly. The study was in about 500 individuals, and the age range was given as 18 or older. So they don't really say what their upper border was for age. And so if you look at the neutralizing antibodies here, if you look in a neutralization assay with authentic SARS-CoV-2, we have very low titers in the 1 to 20 range for both doses. If you look at pseudotype particle entry assay, we see higher titers, which go to approximately 1 to 60. So these are not very impressive. But they also looked at the T-cell response, and they saw that there is very nice induction of interferon gamma expressing cells per 10 to the 5th ppmcs. If we look at safety, I didn't put the local reactogenicity at the injection site because that's not, I think, too helpful. But they had a relatively high percentage of people who developed the fever. You can see that here in the high dose, it's 32%. In the lower dose, it's 16%. But I mean, that's not a severe side effect, but certainly something you want to be aware of. Oxford, together with AstraZeneca and Serum Institute of India, have developed a spike-expressing chimpanzee adenovirus vaccine. I think in Serum Institute of India calls it Covishield. AstraZeneca calls it ACT1222. And they have data from a phase 1/2 trial. They went in with 5 times 10 to the 10th virus particles. They tested the prime regimen and the prime boost regimen. Again, this was given intramuscularly. They enrolled about 1,000 individuals between 18 and 55. And what I'm showing you here is the neutralizing activity. They used several different assays, but what we can see here is that they certainly get neutralizing activity after the first vaccination, depending on the assay. That's in the 1 to 20 to 1 to 200 range. And if they give a boost, these titers go up a little bit, but the boost is not very impressive. Which could have to do with vector immunity that is induced. They also looked at T-cell responses, and again, they get a nice amount of spot-forming units per million ppmc. So they didn't really look into CD4, CD8s, but at least they get a relatively nice response, which is something that you would expect from an adenovirus vector. And in terms of safety, they did something very nice. They had a control group that actually got a meningitis vaccine, so you can compare it to a licensed vaccine. And they treated some of their individuals that they vaccinated with paracetamol, and some they didn't treat. It seems that the treatment didn't make a difference, but you see that the safety signal in terms of systemic side effects is a little bit higher in their vaccine, which again makes sense. Most of what we see is things like headache, fatigue, so nothing out of the ordinary, but certainly a less safe profile than the meningitis vaccine that they used as negative control. The next candidate is Pfizer. They are teaming up with a German company called BioNTech, which does RNA vaccines. And the candidate that they published so far was BNT162B1. And it's a messenger RNA formulation. Interestingly, they published the B1 data, but they went ahead with the B2 construct. So the B1 construct is an RBD that is expressed in vivo. The B2, as far as I've heard, is a full-length spike protein. But nevertheless, the data that they show here is promising. They went with 3 different doses, 10, 30, and 100 micrograms. They had a lot of safety signals for the 100 micrograms, a lot of reactogenicity. So they dropped that. They didn't use that for a boost. If you look at the neutralizing titers here, that looks pretty okay. For the low dose, they get about 1 to 160 after the boost. For the higher one, 1 to 260. So there's actually not that much difference. And they didn't assess the highest dose anymore in the boost because, as I said, they had a lot of reactogenicity. And this is basically shown here. So you see the safety profile after the prime and after the boost. And the 100 microgram dose doesn't look very nice, while the 10 and 30 microgram doses look quite okay. Again, the majority of the side effects here are things like fever, fatigue, headache, basically what you would get after your innate immune system is stimulated. That's something that these RNA vaccines are actually doing. It's more related to the LNPs that they are delivered in than to the RNA, but that's something that people see consistent. And then we have Moderna and the Vaccine Research Center. They have their mRNA 1273, which also codes for a full-length spike protein. And they just released data from their phase 1 trial. They had 2,500 and 250 microgram of RNA that they used in a prime boost regimen. Again, they don't get much of an induction of neutralizing antibodies after the prime, but they have substantial titers after the boost. Again, what I'm showing you here are different neutralization assays. On the upper side, on the upper panel, you see pseudotype virus neutralization. So they go into the 1 to 500, 1 to 300 range, but that also holds true in their case for a plaque reduction neutralization assay with the actual virus. And that's what you can see here. Those are the 2 lower doses after the boost, and you see substantial neutralizing activity. And there's basically no people that don't respond to the vaccine, which is great. Again, this was just published. They also looked for T cells, and in this case, I'm showing you the 100 microgram dose. There doesn't seem to be a lot of a response in terms of CD8 T cells, but they get a substantial response in terms of CD4s, which basically goes hand in hand with the antibody response. And left and right here, just the S1 subunit and the S2 subunit of the spike protein. Again, they did not move ahead with the highest dose, which was 250 micrograms because of reactogenicity. You can see here, again, we have very similar safety profiles as for the other vaccines. There's injection site pain. There is basically headache. There is fatigue in some of these individuals. And what is interesting here is that the symptoms actually got worse after the second vaccination, as you can clearly see here. But in general, there's at this point nothing to be really concerned about. And then we have Novavax. This data came out today in medRxiv. They had a press release recently with PowerPoint slides where they also showed it. This is very interesting and very encouraging data as well. This is from phase 1. They're currently in phase 2. And they tested a spike protein expressed in insect cells that is then purified from the membrane and forms nanoparticles or rosettes. And they used 25 microgram of that unadjuvanted, or 5 micrograms with matrix M, or 25 micrograms with matrix M in single shot or prime boost regimens. And again, what you can see here are the neutralizing titers. So this is again with actual SARS coronavirus 2. If you don't add an adjuvant, the neutralization titers are pretty disappointing. But if you add an adjuvant, these titers are amazing. So we go into the 1 to 4,000 range, basically, with the 5 microgram dose. It doesn't seem to help if you add more protein, more antigen. So they seem to be equivalent. And even after 1 dose with adjuvant, you get into the 1 to 80 titer range. So this is pretty substantial and goes well with their non-human primate data. In terms of T-cell responses, we see the same as for the other vaccines. There certainly is, depending on the group, a CD4 response that shows up. I don't think that they looked at CD8s, but consistently we see now that inducing CD4 responses works very well for the spike protein. That's also what Alex Sette and Jane Crotty have shown for natural infection. In terms of reactogenicity, of course, if you add an adjuvant, you get some side effects, some reactogenicity. It's very similar, again, to the other candidates. Headache, in some cases, fever, there's actually not that much. There's just, I think, 1 person in the higher dose here. But in general, again, it looks pretty good and looks pretty acceptable. So just to sum that up, we have now clinical data from 5 studies, 2 with adenoviruses, 2 with RNA vaccines, and 1 with a recombinant protein vaccine with an adjuvant. The vaccines that are based on the adenoviruses, if you give them once, you don't get much much neutralizing activity. If you give them twice, it's actually a pretty good range, somewhere in the 1 to 20 to 1 to 250 range. The RNA vaccines seem to do very well, but you seem to need 2 shots. One doesn't seem to be enough to get a good neutralizing titer. And Novavax data is, in my opinion, I haven't read the preprint in detail, but I looked at the data. It's relatively outstanding with reaching titers in humans in the 1 to 3,000, 1 to 4,000 neutralization range. Again, the neutralizing titers have to be taken with a grain of salt. These are all different assays that are set up in different laboratories. So you can compare them, but, again, with a grain of salt. And so in conclusion, vaccines have been made in record time. There's many candidates in the pipeline that makes me very happy. There's many candidates that are already in the clinic, which also makes me happy. I'm saying this makes me happy because there's not a single vaccine producer that can actually meet the demand of the market. And also, if some of them fail, there are alternatives. And so it's really, really good to have a high diversity here and have many candidates. We see pretty encouraging results from preclinical models in phase 1-2 trials. But there's, of course, open questions that remain. None of this means anything. The proof is in the pudding, and the pudding in this case are phase 3 trials. That's where we will learn about the actual efficacy and the safety. The reactogenicity for many of these vaccines is on the higher side, specifically local reactogenicity, which might not be a problem for adults, but what I'm asking myself is how these vaccines will do in children and if they will be acceptable in children. And of course, we don't know how the protection looks like, if this will lead to protection from disease, which I'm relatively hopeful about, or if this would also lead to protection from infection, which I'm less hopeful about. And infection, if they're infection permissive, they might actually also be transmission permissive to a certain degree. So we have to keep that in mind. And the last point that I wanted to make here is it's very important. And, people are trying to suggest to do that, but we cannot cut corners in terms of safety or efficacy. Even if other countries like Russia are trying to have a vaccine on the market in October, even when China allows the use of their adenovirus vaccine in the Chinese military already, I think we really need to wait what the phase 3 trials tell us, and we need to rely on the FDA to make a judgment call and only license vaccines that are safe and that work. Even if they are not perfect in terms of efficacy, we want to see the data first before there's vaccine rollout. There was a lot of discussion about that lately, but I think that's really a very important point. I think it's good to fly this ship with warp speed, but it doesn't help if you fly it with warp speed into a planet. So I think we have to be careful about what we do and how we get this license and how we get this to the population. And with that, I just wanted to thank my team here. We're doing a lot of work on COVID. There's a lot of people involved. Yeah, with that, I'm happy to answer questions. I hope there is time left.
Florian, that was amazing. Yes, you have a ton of questions. Okay, so here we go. So we've got 2 questions about inactivated vaccines. Adam, okay, yeah, okay, Adam's being turned on, and David also had that question. So can you unmute Adam and ask away?
Hi, can you hear me?
Yeah.
All right. Florian, great talk. And also thanks for your service on Twitter over the months of informing us all about things that are going on. I'm trying to figure out why the sort of inactivated viruses have not been sort of funded and by the US government and pushed. Is it really just a production thing, or is there something else going on? And can you solve the production problems with enough resources? And also, do you know much about what China's inactivated viruses vaccines look like in terms of their efficacy and safety?
Yeah, very good questions. So there's a few things. The first point is that it has been shown that formalin inactivated SARS coronavirus 1 vaccines in animals can cause enhanced disease. Beta-propiolactone inactivated vaccines did not do that. But I think that's one of the reasons why Western companies are basically shying away from using those. The other problem is that you need a biosafety level 3 production facility for making them. And that's not so easy. There's not that many of those. I think that's another reason that can be circumvented by using an attenuated virus for making inactivated vaccines. But again, that is also complicated. So I think those might be the reasons. And China just has these facilities, and that's what they are using, which makes sense. By the way, India, a couple of companies in India are also starting to develop inactivated vaccines.
Awesome. Hey, David, you had that question, and it looks like you covered that. Can you ask your other question about spike protein? So, yep. Okay, you're up, David.
Yeah, hi, Florian. Great talk. Thanks so much for all the work you're doing and for your great Twitter feed. I just wanted to ask you, you were saying that the spike protein is hard to make as a recombinant protein, but the RDB works well. And I'm just wondering what we know about RNA vaccines and how the spike protein is transcribed and translated, and whether the epitopes are preserved and so on. Maybe we know that from neutralizing antibodies from animals and from clinical trials.
Yeah, hi David. That's a very good question, and I don't think there's a systematic review of that. What I have seen, if you look, for example, at the Pfizer data, they expressed the trimeric RBD, right, in vivo with their RNA vaccine. And if you look at the ratios of binding antibody versus neutralizing antibodies in their vaccinated individuals compared to the controls from natural infection that they had, the ratios are way worse, right? So you get a much higher proportion of neutralizing antibodies after natural infection, it looks like. But I don't think there's really a good analysis of the in vivo folding and the in vivo expression levels if you compare RBD and spike.
It just seems like this is another risk of working with such an untested technology and putting it at the forefront of all the vaccine development.
Well, that's correct. It's hard to control, right? But I mean, again, the titers that you see, the neutralizing titers with the RNA vaccines are relatively high. So it's hard to say, but you're right. I mean, we don't have experience with those vaccines, right? So.
Thank you.
Florian, no one ever talks about adjuvants, but, you know, this is in my mind, this is this is what it all hinges upon. like the Novavax showed a 10-fold higher neutralizing antibody titer with adjuvant versus not. And their adjuvant is proprietary. I mean, and there's like 30 different adjuvants. How do we figure this out? It's such an important piece, and it seems like such voodoo.
I think it was even 100-fold higher almost, right? Yeah, that's really hard. I know that GSK said that they would make ASO3, which is also a pretty good adjuvant, available to vaccine producers, and they're partnering now with different vaccine producers. I think Seqirus is doing the same with MF59. So, there is access to additional adjuvants that have a proven track record, but it's complicated. And I'm actually not sure that how the supply for saponin, which is a component of matrix M, it's Novavax, that Novavax is using, how the supply for that looks like, right? If you have to make millions and millions of doses, not sure if there is enough available. So it seems the adjuvants are very important, and I hope there's going to be enough of them. Some of them are easier to make than others, but yeah, it's a very important point.
Yeah, okay, now we've got Louise. Go ahead, Louise.
Hi, great talk. I was curious what you think of the COVAX vaccine, which is based on a synthetic peptide. I don't know if you've heard much about it, and how it maybe compares to the other existing vaccines. Thanks.
Thanks for the question. Yeah, I haven't heard of that. There are a number of peptide-based vaccines in preclinical studies. I'm usually not a big fan. I'm an antibody person. Antibodies that are potent usually bind to conformational epitopes. It could work, but it's nothing that I would have seen that really stood out so far.
Well, so Florian, I've got the details on COVAX. So it's a soup of things. You do give them the full RBD, but their trick is they add peptides that should be good MHC displayers for T helper and CTL ligands.
Do you know at which stage it is? It's probably preclinical, right?
I think it's,
No.
I think it's phase 1.
Yeah, phase
Okay.
1, 2.
Yeah.
Yeah, I haven't looked into that, to be honest.
Okay, thanks.
Okay, next up we've, Ava, can you go ahead and ask your question?
So thank you so much, Florian. I did type my question in the question and answer, so maybe you haven't seen it. So I love your presentation, very informative. You ended up talking about safety and efficacy, and I may be a bit naive. I see the absolute relevance of safety, but when it comes to efficacy, given that these vaccines are being produced now before we know whether they're fully efficacious and the need that we have, if something was, I don't know, 50% successful, wouldn't that be something better than nothing until we get a vaccine that really gives us 100% protection? Is this very naive way of thinking?
No, and I don't think that we will end up with a vaccine that gives you 100% protection. Vaccines typically don't do that, right? And I think something with 50% would be much better, at least in the short run, than something that has, no help at all. But you still need to wait for the efficacy data for that. You don't want to take a vaccine with unknown efficacy and start to vaccinate people and tell them now they're vaccinated, right? Because they may engage in very risky behavior, assuming they are protected. And what happens if the vaccine doesn't do anything? Even if it's safe, you put those people at risk, right? So I think that's important to keep in mind. But I completely agree. We're not looking for the perfect vaccine. We're looking for, 50% efficacy would already be nice.
Thank you.
Let's see, Nikki, can you please?
Nikki, unmute yourself, please.
Hi, can you hear me?
Yeah.
Yes.
Yeah.
Hey, thanks. Thanks to everybody, Florian and the entire team. This is fantastic what you guys have done. My question was about the last conclusion slide that Florian showed us. And there he had one point where he made, which he said, about adults versus how it might work in kids. If he could just elaborate, that'll be lovely.
Yeah, of course. What you often see with vaccines is that you have a lot more reactogenicity in kids, right? For example, there was the case for the cellular pertussis vaccine that then had to be switched to the acellular pertussis vaccine. Because of reactogenicity in children, we switched whole inactivated influenza vaccines to split vaccines, right? And so if you see safety profiles in adults that already show a lot of reactogenicity, that might be stronger in kids. And the question is, is it at an acceptable level in children or not? And this can be tested. I'm actually not sure what the vaccine producers are planning for the phase 3 trials. Typically, you do age de-escalation. You would basically start in healthy young adults, and then you would go down, right, in terms of age. But again, it's not clear if you would see a safety signal there that's unacceptable, and you might end up with a vaccine that is licensed in 18 plus, but not in people younger than 18, for example. For flu vaccines, for example, for flu mist, that's only licensed above the age of 2, because when they tested it below that age, there was a safety signal, right? So this happens frequently. But again, kids are usually more prone to reactogenicity.
Very important point. Thank you, Florian. Thanks.
Awesome. So next we're going to call on Ronnie, but also Vincent and Victor. Can you also please raise your hands?
Hi, everybody. Thank you very much for hosting that seminar. And Florian, really appreciate the insight here. Can you talk a little bit about the temperature in which we have to keep the vaccines in order to be able to distribute them? My understanding, some of them have to be distributed in very cold temperature. I was wondering if you actually have the specific knowledge of what temperature they have to be distributed at, each one of them. And is that really a problem, or is that just something I know we can get over? And then if you have a chance, we can talk also about the risk of mutagenicity of the virus and the risk it will be able to escape at least some of the vaccines.
Yeah, I'm happy to address this. So I think right now the formulations are all at 4 degrees Celsius, right? So I'm not 100% sure what the stability over time is at higher temperature, but of course that's a very important point for specifically low-income countries, right? For high and middle-income countries, it's less of an issue. So I'm not really sure what the temperature profile or the storage conditions need to be for those vaccines. You could imagine that protein vaccines that consist, for example, of the RBD, which seems to be a very stable protein, could be stored at higher temperatures than, for example, a spike protein-based vaccine, which seems to be relatively labile. On the other side, Jason McClellan has made a very stable version of the spike protein too, right? So this is something that probably is investigated right now because people have that in the back of their minds. But right now, I think the candidates that we see in the clinic are probably candidates that need to be stored at 4 degrees Celsius. To your second question, we know now that there is neutralizing epitopes on the RBD, on the MTD, and probably also in the S2. So what I would assume is, in terms of virus antigenic drift, vaccines that are based on the full spike are safer and might be, might give you protection longer than vaccines that are based on RBD, just because RBD is much smaller and there's probably fewer epitopes and they might change, right? But in general, I'm not too worried about the virus mutating. I know we have the bad example of influenza, which antigenically drifts all the time, but even for other RNA viruses, that's not what typically happens. And the other problem that the virus has, it has a very large binding interface with the receptor, with human ACE2, right? And it needs to maintain that. If it changes that binding interface, the virus might become less transmissible or even attenuated. Flu, for example, doesn't have that problem because it binds to a very small sialic acid in a receptor binding pocket, and everything around can be changed without changing the affinity, right? So I'm less worried about that, to be honest.
Thank you.
Awesome. Next up, we have Vincent.
Vincent, you're allowed to talk. Go ahead, please.
Great. Thank you so much, Dr. Krammer. Amazing talk, and I'll throw in my thanks as well for all your insights here and on your fantastic Twitter feed. I was wondering, how optimistic are you about the potential to get to sterilizing immunity with sort of this initial crop of vaccines? How likely is it that some of the sterilizing immunity that you've seen with the J&J and the Novavax animal studies and non-human primates can be replicated in humans, and why or why not? And then if I can toss in a second one, if you have a chance, I was wondering if you had any thoughts as to the likely durability of protection following vaccination. I recognize it's still early days.
Yeah, very good points. So I do not expect that any of these vaccines give you sterilizing immunity. I do not think that that is an issue. I think you might get an asymptomatic upper respiratory tract infection in some cases. It might not lead to disease, and it might not be enough virus to transmit it on. I think it depends on the titers that you get. I'm not too worried about it, but I'm not very optimistic that we get complete sterilizing immunity. We're not even sure we get that with flu vaccines, even if they're matched, right? I'm not too optimistic, but I'm also not too troubled by that. Then the other point was, how long-lasting is the immunity going to be? So what we're seeing now is that for the natural infections, we get a relatively normal immune response. I know there are some reports out there about rapid waning, but if you actually look at the data, it just looks like an immune response that you would see for other viruses too. For the vaccines, it's less certain how that pans out, and some of them might actually induce longer lasting immunity than the actual infection. But again, I don't see that as a big deal. So I grew up in Austria, in Europe, and we need to get vaccinated with TBEV, tick-borne encephalitis vaccine. And you need to get revaccinated every 4 years or so because the titers go down. And you could have a similar scenario where you have to get revaccinated with the same vaccine from time to time. But, that's not a problem. And we do that for other vaccines too, right? You sometimes need to get a refresher for a Hep A or Hep B or even tetanus or something like that.
Thanks, Ray. If I could just follow up quickly, what would you say is a realistic expectation for how protective these might be in terms of maybe it's not sterilizing, maybe preventive? Just where do you think you alluded to protection against sort of different levels of protection. What do you think is realistic for the first crop of vaccines?
I think protection from disease is pretty realistic. But again, I like to look at data, so I'm waiting for the phase 3 trials. They will tell us.
Thanks again.
Awesome. So we'll have Victor next, but first, Alex, Roman, and Rachel, can you please raise your hands? Okay, so Victor, you're up.
Okay, so my two questions. First was your thoughts on intranasal versus intramuscular. And the second is your thoughts on if several vaccines get approved now, what are your thoughts in terms of other companies continuing to develop vaccines in the future?
Yeah, two very good questions. So I think mucosal immunization, if you deal with a respiratory virus, is always a good idea. But we have seen with flu mist, for example, that it's also in a way problematic because sometimes you see protection, but you don't measure very robust immune responses. But in this case, you would need to go through a proper phase 3 without a correlate of protection anyways. So I would like to see more live attenuated vaccines that make it into the clinic. They might have an advantage. So that's my comment there. In terms of having vaccines on the market that are licensed, if they work well and if they are safe and they have little reactogenicity, and there's a couple of companies that make them, I think vaccines that come a year later will have it really hard. Will have a really hard time to make it to the market, and I'm not sure if all of these candidates that are in the pipeline right now will even make it to clinical trials, right? Once you have positive results from 2 or 3 good candidates, you know, some of those pipelines might try out. So not sure how it's going to pan out. If the first candidates are acceptable but not really great, I think a second generation vaccine is needed. If the initial candidates that come through are good and acceptable, then it will be a tough time for basically vaccines that are not that far in the pipeline yet.
I do think you made a good point earlier, though, about, you know, we're going to need a lot of candidates to tile the world. So, you know, I can imagine that, you know, like the Chinese trials would probably be relatively unaffected by results in the American market. It's just my naive take. But
no, that for sure. I think you have to keep that apart, because if you think about Europe and the US, it's very easy to, if you license a vaccine in the US, it's probably very easy to get the same vaccine licensed in Europe. Mm-hmm. I'm not sure that Chinese vaccines will be licensed in the US without having clinical trials here and maybe even production facilities here, right? So the markets will be separate in a way, right? So I was more or less talking about Europe and the US.
I mean, even for Europe, though, I mean, it just seems like production is going to be such a problem that I can imagine they'd want to go full speed ahead with a lot of different candidates. But, okay, let's see.
Roman?
Roman.
Roman, go ahead, please. Unmute yourself and ask away.
Yep. Hi, Florian. I have a question regarding the inactivated vaccine. There was a paper showing that virions that were treated with beta-propiolactone were in the post-fusion conformation. So do you think that this might be an issue for neutralizing antibodies because RBD is gone in this case?
Yeah, it could certainly be an issue. I mean, the Sinovac studies showed that you can induce neutralizing antibodies. In the non-human primate model, even after 3 shots, they were not very high. And that could be one of the issues there. I guess it also depends on not just the inactivation, but the whole production process. You could have different ratios of post-fusion versus pre-fusion spike, depending on your production process. But that's certainly something important to keep in mind.
All right.
Awesome. Alex, can you unmute yourself?
Thanks a lot. So I have a question about the fact that the SARS-CoV-1 vaccines that appeared many years ago were sidelined because they seem to cause immunopathology in different animal hosts. Is there any evidence that that could happen with some of these SARS-CoV-2 candidates?
So you can exclude it. You have to be careful. I mean, there were actually SARS-CoV-1 vaccines that went into the clinic. There was a DNA vaccine by VRC, and then there was an inactivated vaccine that was produced by Sinovac, actually. They went into clinical trials. So not all of them were sidelined, right? We have to be careful with enhanced disease and SARS coronavirus 1. There's a lot of data. There's a lot of inconclusive data. There's talk about antibody-dependent enhancement. There's talk about enhanced disease, but not necessarily antibody-dependent. There were weird findings where viral vectors were inducing an immune response, and then when ferrets were challenged, they got basically liver disease. But then in other studies, the vaccines worked very well, right? So I think it could happen with SARS coronavirus 2 in animal models as well. It doesn't seem that it's so easy to induce that type of response. And I guess that's also why it's very important to do proper phase 3 studies and really look into the safety of these vaccines.
Yeah, Kiko had a good piece on that. Wasn't the vague consensus that was mediated by a T helper 2 response, the lung injury from the SARS one?
Yes and no, but there were some papers that reported lung injury. There were other papers that reported other problems, right? there are papers that report antibody-dependent enhancement in vitro, but the same antibody is protective in vivo. There were reports of ADE in vivo. In my opinion, it's a mess.
Yes. Okay, Cecile and Rachel, can you please raise your hands?
Cecile, go ahead. Unmute yourself.
Hi, great talk, Florian. I was just wondering if there was a good hope of generating an immune response in older adults and the problem of immune senescence. Obviously, it's the highest risk group here.
Hi, Cecile. Yeah, that's a very good question. So I'm not too worried about safety in older people, but of course I'm worried about immune responses, right? We know that we have a lot of trouble with flu vaccines to actually induce neutralizing antibodies or high titers of neutralizing antibodies in older people. And there have been formulations specifically for that age group to ensure that they respond well. So it's not clear if we'll run into the same issue with these vaccines, and that might be platforms that actually work better than other platforms in older people. But there might be issues, and the other problem is, I think that's largely ignored. If you look at influenza, you need much higher titers to protect old people than the titers that are needed to protect young people, right? So even if we reach antibodies thresholds in old people that are protective in young people, that might not be enough in that age group. But again, I'm worried about it, but we'll have to see how that goes. And some of these vaccine producers actually include people up to age of 75 or 80 into their phase 3 trials. So this is something that we should know about relatively soon.
Great, thanks.
All right, Rachel, go ahead.
Hiya, good evening. I'm almost a lay person and I learned a lot tonight. Thank you very much. I have understood now that disease protection might be possible and infection protection is probably not possible. So I would be very interested to know your thoughts about the BCG vaccine protection, please.
Okay, so I'm in general a little bit skeptical about that. That is basically, I guess, based on innate immune response, activation of an innate immune response that then would protect you from getting severely sick or getting infected. It's relatively unspecific. I would assume it's also very short-lived, and probably the effect size is very low. So I haven't looked into it in detail, but I would not I'm not a big fan of that strategy. Specifically, if you can actually make specific vaccines in a very short time frame.
Thank you.
All right, Hal, go ahead, please. Unmute yourself and go ahead.
Hello, can you hear me?
Yes.
Yes.
Just curious, you spoke a little bit about the probability that the vaccines will protect you from transmitting the virus. Obviously, there's a difference between complete sterilizing immunity and protection against transmissibility. How do you think about the odds that one of these vaccines will protect against transmissibility, or, if it's not black and white as it'll protect everybody, what portion of people do you think it will protect from transmissibility?
That's hard to say, but it's a good point. I mean, it's a gray zone, right? You might get an upper respiratory infection, but very low titers and for a very short time, which reduces the risk of transmitting the virus probably very, very significantly, right? But it's really hard to predict that right now. I give it a good chance that that all these vaccines certainly dampen transmission. But, I wouldn't wonder if we hear of cases where a virus was transmitted among infected individuals. We just put a paper on bioRxiv a few months ago of a nosocomial outbreak of influenza in a completely vaccinated population in a hospital in New York, right? And so they had basically very little disease, but they were still spreading the virus.
I guess if I could just follow up quickly, because there's been quite a few reports about asymptomatic people spreading the virus as well as symptomatic people. So I'm just curious why you think, or what gives you confidence that you'll be able to reduce transmissibility without reducing infection?
So, you know, infection is a category, but actually you have different titers in different people, right? So if somebody has an immune response, a strong immune response, you might still be susceptible to infection, but you might just have very low titers or for a shorter period of time. I think we have to be a little bit careful with asymptomatic transmission. There's two things there, and I think the WHO didn't do a great job communicating that. There are people who are asymptomatic, they transmit the virus to somebody else, and then they become symptomatic. So those are pre-symptomatic transmitters. And then there's people who are asymptomatic, and they might also transmit the virus. But as far as I understand it, and as far as the data that I've seen, those people who stay asymptomatic are not very likely to spread the virus compared to pre-symptomatic individuals who are, you know, there's many cases where that has been shown, right? So I think we have to keep those two things apart as well. And that might also have to do with viral load in a way.
That makes sense. Thank you.
All right, Nelly, go ahead, please.
Hi, this is Neeltje van Dorenmalen. Thanks for the talk, Florian. That was really interesting. I was wondering, because you already alluded to this at the beginning of your presentation, what do you hypothesize the correlates of protection are going to be both for the lower respiratory tract as well as for the upper respiratory tract?
I'm an antibody person.
So you're going to say neutralizing antibodies?
I think we're going to end up in a similar situation as for flu, where it's just easier to prove antibody-based correlates of protection. While if you look at CD8s, there might also be a correlate of protection there that might even be independent, right? I think it's just easier to work to establish that for antibodies. So that's my guess that that's something that we will get relatively soon. But I'm certain there's also contributions by T cells, right? Yeah. By the way, congratulations to the paper. I really liked it.
Oh, thank you. I appreciate that.
Awesome. Thanks, Neeltje.
We still have 15 questions. We are enjoying as much as the audience is, but we just want to make sure we are still okay with time as far as you're concerned. We really appreciate if you could answer a few more questions.
I do have a conference call in 15 minutes, but I have time until then.
Okay.
Thank you. We truly appreciate it.
Try to speed it up. I'm just going to read out a question from Anonymous. When do you think we'll start to get results from phase 3 trials? And when do you think we'll have conclusive results from phase 3 trials?
I guess that we'll get results about efficacy relatively quickly, just because there is a lot of transmission in the United States. So there's a lot of trials that are taking place at different locations in the US. You know, the trial results the results that you will initially get are based on relatively short-term immunity. If you want a result on how protective the vaccine is after 1 year, you need to check after 1 year, right? So I guess there will be multiple analysis, interim analysis of these phase 3 trials, and I'm assuming that there will be also kind of co-review of those results by the FDA. I'm not sure how it will actually go through regulatory agencies, but I assume that we'll have pretty good data in late fall or you know, around December, maybe before that.
Let's see, piggybacking on that, Alex Guzman, can you quickly review what could go wrong in phase 3?
Oh yeah, sure, many things. What could go wrong is that you don't see efficacy, right? If you see 20% efficacy, I don't think that would go forward, right? 50%, I'm not so sure it might. If you see severe adverse events that are really caused by the vaccine that you didn't see in a few hundred people, but you see, you know, 1 or 2 or 3 of them in a few thousand people. I don't know, neurological issues, autoimmune disease. I think something like that could kill a vaccine relatively quickly.
Yeah.
So many things that can go wrong.
Yeah, and I'll just chime in that these things have happened before. Many vaccine candidates have looked spectacular in phase 2 and then were a complete disaster in phase 3. The herpes simplex, Staph aureus, RSV, et cetera, et cetera.
So just to jump on that, we had a case in 1976 with the Fort Dix swine flu outbreak, where 6 people got sick with swine influenza. It was determined that this looks like the 1918 virus, and all of a sudden there needed to be a vaccine very quickly. Many people were vaccinated, and the vaccine caused Guillain-Barré syndrome, and the virus never spread. So that's kind of one of the big disasters that happened. But there are other examples as well. So many things can go wrong.
Yeah.
All right, just to speed things up, we'll read out more questions. I'm reading out a question from Buzak Uzail. What can be the unintended consequence of an mRNA vaccine?
Yeah, that's a good question. We don't know. It's new.
We will find out.
I think the most likely things are things that we are seeing right now, which is basically reactogenicity, induction of strong innate immune responses, fever, things like that. But we just don't know.
Historically, non-human primate vaccine trials have not been wildly predictive of human response. How optimistic are you about the results that we're seeing in NHP now, that it'll hold up?
Well, there's differences for different vaccines, right? One problem is that if you test an F5 vaccine in an NHP, it might work better than in humans because they don't have pre-existing immunity to F5, right? So that might be a factor. I think it's a pretty okay model for SARS coronavirus too. But again, it's an animal model. Typically you do multiple animal models, and many of the vaccine candidates actually go through multiple animal models. The Moderna vaccine, for example, you know, looks good in multiple animal models, others too. So I think you can never 100% predict, but if you see good protection in several animal models, it's usually a good sign.
All right, I figured Chris's question. Is herd immunity important for a COVID-19 vaccine, given it only harms a fairly small proportion of the population? Surely a vaccine that stops severe illness but may still allow transmission be enough to get back to normal?
Probably, and I think that's what we will get to some degree, right? But again, if you have a vaccine that dampens transmission, you might actually get very little transmission of the virus in the community. Now, I think we have to be careful anyways. The vaccine is not going to eradicate the virus. This is going to be human coronavirus 5. This is not going away anymore. At least I don't believe that. I mean, I think if a large proportion of the population is vaccinated, there will be much less transmission, and that will actually also, you know, passively protect people who cannot get vaccinated or don't mount a good vaccine response.
All right. Lauren, do you want to read the next question?
Aaron asks a fascinating question about what do you think will be what will happen to the people who were in vaccine trials that were not very protective? Could that adversely affect their fate going forward, or should they be just fine?
That's really hard to answer. Likely with all these spike-based vaccines, if you get neutralizing antibodies but not enough, or if it turns out that the vaccine doesn't work too well, you probably get vaccinated again with another vaccine afterwards. If you're lucky, and we know that from a lot of studies with prime boost regimens where we use different vaccines in sequence, that might actually even give you a better immune response. But of course, there are immune mechanisms. We know that from influenza, like original antigenic sin, where you could be primed and then you make a not that great immune response, even if you get vaccinated with a good vaccine afterwards. Hard to predict. I would say it's not likely that this is going to be problematic, but obviously it's something that needs a follow-up. And usually those phase 3 trials have pretty long follow-ups with their subjects.
All right, here's one for you from Bryson. If you were allowed the opportunity, okay, I missed that. Did you answer that? No.
It's basically if you had to pick a trial.
You had to pick a trial. Which one would you sign up for? Pfizer, AstraZeneca, or which one would you sign up for?
So as I just mentioned, I'm very much into prime boost regimens. I would probably take the AstraZeneca Oxford vaccine as a prime and then get the Novavax vaccine as a boost. They wouldn't allow me to do that, so for now I'm staying away. To be honest, I want to see the phase 3 data first. Mm-hmm. I'm not going to sign up for a trial before I see that, probably.
Mm-hmm. And I've got a related question. How likely do you think it is that will be recommended to get like a combination, maybe an injection and a nasal for mucosal immunity?
I mean, this is very complicated, right? Also what I just mentioned, to get a prime boost regimen, because you actually need to do trials to show that that works as well, right? So it would probably take a very long time to develop or to show that this works, right? Of course, in a way, you can stop people from getting multiple vaccines, right? So people could still do that. If it works, if it's better, if it's worse, that's then a question. But hypothetically, if you really see a combination that works very well in a certain part of the population, let's say older people, and there's really kind of evidence that it might be helpful, you could do a trial and establish that, and then there could be a recommendation. But I think that's complicated.
In present, a couple more questions before we.
Yeah, maybe just one or two, and then let's let him go.
Is there a process in the US known for deciding who gets the vaccine first, and will it be published?
Well, I think usually for the rollout, the CDC would do that. I'm not 100% sure. And what I miss in the public right now is a discussion about that. I would like to see a very lively discussion about how we do that once we have it, who gets it. I would assume healthcare workers and probably high-risk people. But I would like to see a discussion about that. I want to see what the public thinks about that, and I think there should be a dialogue. And I'm not so sure if there is a good plan for that right now. Probably there is, but still it would be nice to kind of see a dialogue about that.
Mm-hmm. All right, Strata, maybe one more, and then I've got a quick one, and let's let it go.
Will phase 3 trials include tests of patients who have coronavirus antibodies to determine the effect of vaccines on those who have pre-existing antibodies? This from anonymous attendee.
So, I don't know the details of the trial design, but for example, the Oxford study already included that. They had people who had baseline antibodies. So I'm pretty sure that at least some of the phase 3 trials will include that as well. It's an important question, right? Because there is a good proportion of people in some countries and in some cities that have already immunity, and you wouldn't get tested before you decide if you get the vaccine or not. So that needs to be answered.
And Florian, I just want to leave you with one that I read. I read a lot of panicky articles about the global glass shortage, how there were only 200 million bottles for vaccines, and if we're going to need 16 billion, what do you think about this?
Yeah, it's a challenge. I think CEPI is doing a good job of organizing all of these things. There's basically a global push to organize all of that and make it happen. There might be other ways to package these vaccines. Maybe it doesn't have to be I don't know what is called. There's a certain grade of gas that you need to actually package it, but there might be alternative packages. So, that's one of the challenges. There's many challenges in terms of how do we package that? Is there enough material, syringes, and so on and so forth. So we'll see how that pans out.
Yeah.
I wouldn't panic about it, but it's certainly something that's important.
I've got enough to panic about.
The questions that we did not cover, we'll probably email Florian and try to post the answer along with our recording, because I guess as much as we'd love to continue talking, he probably has another call.
Yeah. Yeah, so thank you so much, Florian, for spending time with us today and everything you do.
Yeah, thanks for having me. It was fun.
Yeah. And thanks, audience, for all the good questions. And we'll see you next week.
Thank you.
Bye.
Bye-bye.